I've been asked by Susan last month to give a quick overview of what is epigenetics. You might have heard of that at different levels, especially after the Human Genome Project was completed. New focus, new targets emerged, and epigenetics was one of the new horizons of the genetic research. So you will see here something that may look a little complicated, but it summarizes in a very simplistic way some of the major changes that allow epigenetics to regulate the function of our DNA or our genes. So by the end of the presentation, I hope that you will become a little bit more familiar with all these little modifications, the methyl group, the histone modifications, and so on. So let's start with the most simple things, the definition. So epigenetics comes from Greek, as most of the scientific terms, and is a composition of epi, which means above, and genetics, which of course means origin, creation. So it indicates everything that is above genetic, above what is determined by the genetic sequence. So it regulates the genetic expression, so the expression of the genes. Genes produce proteins, and proteins are the ones that exert the different functions in our cells. In order to regulate that, it does not change the gene sequence. Now, we know that if you change the sequence of a gene, you have a mutation or a variant, and that, of course, has an effect on the protein structure, function, localization, and so on. With epigenetics, we have a regulation that does not change the gene sequence. That is why it's above the gene. And it is above because it is mediated by chemical modifications. So there is an addition and removal of chemical groups to the DNA molecule or to molecules associated with DNA. And by adding or removing these chemical groups, you will have the result of modifying the level of activity of the gene. So how much that gene is expressed, so how many products the gene produces, how many proteins, but also modulating the time of the expression, the localization of the expression, as we will see in the next slide. The very important feature about epigenetic modifications is that they are reversible. So whatever you add, you can remove. So that means that if a gene is turned off, you can turn it back on. And that is very important because, as we will see, epigenetic modifications are very fluid, and they are a sort of an interface between our genome, so the DNA sequence, our genetic code, and the environment, because the environment may not modify in a very finely regulated way our gene sequences. We know that there are environmental factors that can cause mutations, like cigarette smoking, but there is also this interface where the environment, like diet, drugs, and other components, may modulate the epigenetic profile. So without causing permanent changes in our DNA sequence, because the epigenetic modifications are reversible. And then, as we were saying, they do respond to external signals. So there are things that we're subjected to, but things that we can determine. Imagine about changing our diet, quitting smoking, having some supplements, or taking some drugs. All these things, if you know what is the target and if you know what is the effect, the chemical effect that they exert on our epigenetic profile, we can modulate that modification. So we can determine the level of epigenetic regulation on certain genes. So these are the key elements that we're going to discuss in this brief presentation: the chemical modifications, the reversibility, and the response to external signals. So let's start with the chemical modifications. The most known epigenetic modification is the methylation. So the addition of a methyl group that you can see here in blue to the DNA sequence, and it works like a traffic sign. Like usually if sequences are methylated, usually because there are exceptions, if sequences are methylated, that corresponding gene is turned off. So you see methylated blue, the gene is off. And why is that so? Because with the methylation, so when we add these methyl groups, then it is not possible to come to read the gene sequence. So the molecular complex that starts the transcription, so the production from the DNA to RNA, and then from RNA to protein, it takes a molecular machine. And if the DNA is methylated, that molecular machine doesn't stick. So it cannot start the whole process that will lead from gene to protein. So adding methyl groups means that in most cases, the gene is turned off. As you can see, this is an external modification. It has nothing to do with the sequence of the gene. It is reversible. You can remove the methyl groups and turn the gene back on. Then there are modifications that affect not the gene, not the DNA, but the proteins that are associated with DNA. In order to allow our DNA to be so coiled, so convoluted, and so compacted, there are several proteins. The most important ones are called histones, and they work as little balls around which our DNA is wrapped, and that helps to keep it condensed. Now, in this case, if you have a methylation of the histone, so these are, again, epigenetic changes, but not on the DNA, but on the proteins that are associated with DNA. If you have methylation of these proteins, then you have a decreased expression. Once again, it means that you don't have the production of protein. But why is that so? In this case, is that if the methyl group is relatively a small group, so if you have those histone tail, so you can see here they're called histone amino terminal tail because they stick out from the complex, these little balls. If you add a methyl group, you don't add a lot of volume. So the different histones, like these balls here, they stick close together. And if they stick close together, the DNA filament that is wrapped around is not accessible to that machine that we were talking about that will start the process of producing protein. If, on the other hand, you add acetyl group, so the acetylation, you increase the expression. And why is that so? Because the acetyl groups are larger. So they create more volume, they space out these balls, they stretch out the DNA, and they make it more accessible to that molecular machine. So it's all about volume here. Methyl groups are smaller, they keep the histones, they keep those balls closer. So DNA is not accessible. Acetyl groups are larger, they make space, they make the DNA accessible. Then there are other chemical modifications, just to give you an idea of the several levels of complexity that are involved in epigenetics. Some of them can have different effects, like phosphorylation or summoylation can increase or decrease the expression depending on where do they bind in the histones. And then ubiquitination is basically a label that says to every protein, this has to be destroyed. So by adding that label to the histones, to those proteins, you remove the histone, you remove the proteins, and you destabilize the DNA. So this, again, is just an example. There are several chemical modifications, all reversible, that may play a role in the expression and therefore regulation of a gene without changing the gene sequence. But there are also other types of genetic regulations that are not epigenetic regulation, that are not just based on adding or removing chemical groups. For example, the histones, we said they are involved in keeping the DNA compacted. Well, the chromatin remodeling. So having chromatin more or less dense means having genes more or less accessible to be expressed. If it's tightened up, the machine, the molecular machine, cannot have access to the DNA. The transcription, the first step is from DNA to RNA. You can see here, DNA to RNA, and that passage is called transcription. The transcription is regulated by several factors. For example, there are regulatory elements in the genes that are not part of what produces the protein, but they are target of several proteins that by binding these regulatory elements may increase or decrease the expression of the gene. So I know this is a lot, but just to give you an idea of the several steps, the several layers where you can have modification, you can have regulation, you can have tuning of the expression of a gene. And going down along this figure, you can have post-transcriptional regulation. So once you have generated the RNA called mRNA, because messenger RNA, it brings the message from the DNA to the protein. That mRNA can be modified. That is why post-transcriptional, so after it has been transcribed. And here you have some examples. Splicing. Pieces of the mRNA can be removed, spliced out. Non-coding RNAs. So there are RNAs that are created to stop the coding RNAs. So they bind the coding RNA and they prevent them from leaving the nucleus and getting to the rest of the cell where they will be turned into protein. So they silence the gene, not by adding again on the gene sequence, but by blocking the messenger RNA. And then there are several other things that you can see here also in the picture. And then we go to the next step, that is translation. Translation is the conversion of RNA into protein, and it occurs at the ribosomes. And then you have post-translational regulation. Again, once you have a protein, it can be modified by addition or removal of chemical groups, and like we saw for the histones, and then allowing or preventing that protein from reaching the secondary, tertiary, or quaternary structure, so that the structure will eventually be functional for the purpose of that protein, the localization, the stability, and so on. So this is, again, something to give you an idea of the complexity of all the regulations that have, the regulation processes that have nothing to do with the gene sequence, but they occur above the genetic sequence, and they're all reversible. They're all modifiable. So, speaking about reversible changes, the epigenetic changes vary during the development. So there are time-dependent changes, but also vary depending on the site where they occur. There are tissue-dependent changes. For example, we know that all our cells share the same DNA. So how is it that the cells in our eye don't produce hemoglobin? Potentially, they have the genes, right? All our cells have the genes to produce all the proteins, but we don't produce hemoglobin or other blood protein in our eye. We don't produce, collagen and in our teeth, or in our stomach, but they're all there. So how is it that certain genes are expressed in certain tissues and other genes are not? Because of epigenetic regulation. Depending on the type of tissue, certain genes are silenced and certain are expressed by applying epigenetic changes. And the same thing is about the time-dependent expression. Certain genes need to be expressed at a certain time in development, and so they're turned on just in that time window. One particular example of the effect of epigenetic regulation is the imprinting. This is an epigenetic mechanism, so again, addition of chemical groups, mostly methylation of DNA, that determines the expression of genes based on their parental origin. So there are certain genes that are expressed only on our paternal chromosome, the one that we inherit from our father, and certain that are expressed only in our maternal chromosome. So how does that happen? because the genes that are not supposed to be expressed are methylated. So they're covered and therefore they're silenced. This is a very delicate mechanism of regulation, you can imagine. And it is based on what? On recognizing which chromosome is paternal and which chromosome is maternal, right? So I'm a man and I have inherited, of course, maternal chromosomes. So when I'm going to pass chromosomes to the next generation, what is going to happen to the imprinting, so to the epigenetic profile of my maternal chromosome? Well, it will be reset. So all the imprinting, all the epigenetic modifications that determine the imprinting are reset in our germline cells. So sperm cells for men, egg cells for women. They're reset, they're eliminated, and then they are reset, like rearranged, based on the gender of the individual. So all the sperm cells from males will have a paternal imprinting. All the egg cells from females will have a maternal imprinting, regardless if they were coming from, if their chromosomes were coming from their father or their mother. And speaking about reversible changes, they can be reversed by drugs, by chemicals, by compounds. There are compounds that have epigenetic effects. So they either increase the epigenetic changes like the methylation and the acetylation and so on, or they remove those groups. So this has been known, and some drugs have been used for many years to exert those epigenetic effects. For example, lithium, valproate, carbamazepine, and they're all involved in neurobehavioral conditions. So there are several conditions for which epigenetic treatments have been approved by FDA: cancer, schizophrenia, anxiety, or post-traumatic stress disorder, diabetic retinopathy. So you can see that several parts of the body, several types of diseases may be affected by dysregulation of epigenetics, and by acting on these epigenetic changes, we may have an effect on these diseases. So we're now close to the end. The drugs are a perfect example of environmental factors, but just to give you a better, probably more impactful idea of how the environment may affect our epigenetic profile, this is a very nice study from 4 years ago, where they looked at an environmental factor, so cold exposure, and the effect of this environmental factor on epigenetic profiles. So they noticed that by exposing individuals before they had children, they were able to induce a metabolic change that was making the offspring more adaptable to that environmental factor. So in other words, they exposed male and female, preconception male in particular, to cold, and they noticed that their generation after several exposure developed a better predisposition to develop brown fat. The brown fat is the one that turns fat not in energy but in heat. So the people that were born from that individual that was exposed were more adaptable. They developed epigenetic changes that made them capable of responding better to that environmental stimulation. And they reproduced that also in mouse models. So we had a better adjustment to cold by mice that were born from males that were exposed to cold. So you see here in the right picture, the heat signal. You see there's more yellow, more red, because these mice were actually better, were more capable of producing heat and adjusting to cold. And that is because their progenitor was exposed to cold. So overall, the mechanism was that exposure to an environmental factor, which is cold, induces permanent epigenetic change, because it's not like you expose the male and that person develops brown fat. No, there is an epigenetic change that is passed through the germline to the new generations and eventually leads to a metabolic adjustment, like higher level of brown fat. So you can see that, as we said, this is reversible, but it's also permanent. So what if you exert a positive effect, that can be passed to the new generations. So with that, just want to summarize some take-home messages. Epigenetic changes may cause genetic disorders, even if the genes are intact, even if you don't have any changes in the gene sequence. Epigenetics modulates the level of gene products, their localization, so the proteins, their localization and their function. So there are several levels on which you can have an epigenetic effect. Neurobehavioral issues, because this is one of the major area of interest, of course, of this organization. Neurobehavioral issues may be caused by epigenetic alteration. So it's not just something associated with the physical trait, something very easy to detect or to measure. So even something more subtle can be affected by epigenetic alteration. And epigenetic modification can be reversed. So they can be controlled. The disorders caused by epigenetic alteration may be treated, may be cured eventually by removing or reversing the alteration. And that's last slide. So.
Wow, thank you. That's really interesting. You really did a great job explaining that. This is part of like why we're trying to push for more research in the area of genetics and epigenetics